Good evening, students. So welcome to Unacademy EAMCET YouTube channel, students. So myself, Dr.Rajendra Prasad, biology educator on Unacademy EAMCET platform, students. In today's class, we'll be discussing about the enzymes topics. In that last class, we have discussed some theory parts. So let us see some multiple choice questions regarding this lesson of the enzyme, students. So before starting the class, as usual, I would like to introduce about our Unacademy Plus subscription. So Unacademy Plus. So Unacademy Plus is for students who subscribe to our courses, students. So Unacademy Plus is for students who subscribe to our courses, students. So let us see what is the importance of our Unacademy Plus and what is the importance of taking the Unacademy Plus subscription. Let us see the benefits of taking the Unacademy Plus subscription, students. So let us see the benefits of taking Unacademy Plus subscription. So premium content. So premium content will be available. So premium content will be available at affordable price. So structured live courses and dedicated live doubt clean sessions will be taking place, students. Structured live courses and dedicated live doubt clean session. After every three theory classes, the fourth class will be a doubt clean session, students. So quizzes. So quizzes also will be conducted by the respective educator, students. So batches. The lecturers will be in batches. You can join the batches of your interest and test series for daily practice papers, full length mock tests. So these all you can get as. Then this all you can get for a single subscription. So of 1 month the trial plan, if you see 1 month trial plan, it is costing around 2,250 rupees, students. 1 month trial plan of about 2,250 rupees. So if you're interested, you can use my code Rajendra Live to purchase this particular Unacademy Plus courses, students. So there is recently one batch was started. The name of the batch is called as EAMCET Rankers batch. In that Rankers batch, you can see the biology educators. We have biology educators, so and mathematics and physics and as well as chemistry educators. So this is the Rankers batch. So if you're interested, this batch was recently started in the December month. So if you're interested, you can use my code Rajendra Live and purchase the 1 month trial plan to get this Rankers batch, join this Rankers batch. Then, so from today onwards on January 11th, so there is one batch was starting. The name of the batch is called as a Telugu batch. So in this Telugu batch, so I'll be taking the biology classes and sir will be taking the chemistry classes and sir will be taking the physics and sir will be taking the mathematics classes. So this is the best batch, students. I suggest you to join this batch so that you can get benefit out of these particular educators. So what is importance of this particular Telugu batch is complete syllabus. So complete syllabus will be covered. Tips and tricks also will be given by the educator, students. Topic wise practice papers and full and mock tests. So this is all you can get for a 1 month trial plan of 2,250 rupees, students. If you are interested, so use my code Rajendra Live to purchase this Telugu batch course. So apart from this 1 month trial plan, if you are satisfied with 1 month trial plan, I suggest you to go for the long term plans of 6 months plan and 12 months plan, students. So these are the cheapest online plans, students. No other platform gives such a cheapest amount for a small cheapest amount. 6 months plan costing only about 10,125 rupees and 12 months plan costing around 12,375 rupees. These are the best two cheapest plans available, students. If you're interested, you can use my code Rajendra Live to purchase this 6 months plan costing around 10,125 rupees and 12 months plan costing around 12,375 rupees. So this is about our Unacademy Plus subscription, students. So let us continue with our today's topic discussion on the multiple choice questions on enzymes topic. So let's see the first question. Say a pocket. So there is a pocket or crevice of an enzyme in which substrate fits is called as. So here they are asking that every enzyme. So if you see the shape of the enzyme. So this is how we represent the enzyme. So every enzyme has. So now this is what we represent the enzyme. So every enzyme has one specific site. Now this specific site is called as active site. Now this is called as an active site. Now this is called as an active site. So every enzyme has an active site. Now this is called as an active site. So this active site. So to this active site, what happens is substrate. So substrate will come and bind actually to this. What happens is substrate will come and bind. So substrate molecules, like these are the substrate molecules. So these substrate molecules will come and bind to the enzyme. So if you see here, now what happens? So like this, to this substrate molecules, enzyme to this. Now you can see here. So enzyme substrate complex is formed. So like this, enzyme react with substrate to form enzyme substrate complex. So furtherly it leads to formation of the product. So like this, so according to the given question, the pocket or crevice of an enzyme in which substrate fit. Now this is called as a pocket. So an active site is called as a, it is actually called as a pocket. So active site is called as a pocket, or it is called as a crevice. So an active site is called as a pocket, or it is called as a crevice. So this pocket, or it is called as a crevice where substrate fits. So that is called as, so based on that, let us see what is the answer. So it is the active site. So based on that, the answer is active site. So active site is the location where it is called as a pocket, or it is a crevices of enzyme. So that is the reason. So answer 1. So active site will be our answer in this case. So let us move to the next question. So that is about the first question. Let us move to the next question. So let us see the next question. So the nucleic acids which behave like enzymes are called as. So if you see the chemical nature of the enzymes, so all enzymes are made up of proteins. So all enzymes are made up of proteins. In that specifically, the what type of proteins they are? So they have the proteins which are involved in the enzymatic activity. They have tertiary structure. So they are having the tertiary structure. So the enzymes which are involved in the proteins, so which are known as enzymes, the chemical nature of enzymes are proteins. So these proteins which are having this enzymatic activity, so they are exhibiting the tertiary structure. But there is an exceptional cases. So there is an exceptional cases in which some enzymes are acting as a nucleic acid. So there is an exceptional cases in which some enzymes are acting as a nucleic acid. So those exceptional cases, best example is, it is the ribozyme. So ribozyme is the exceptional cases in which ribozyme is made up of nucleic acid, that is RNA. Similarly, there is one more enzyme, that is ribonuclease P. So ribonuclease phosphate. P indicates it is phosphate here. So these are the exceptional cases actually. So all nucleic acids, so the means all enzymes, the chemical nature of enzymes are made up of proteins, and specifically in that particular proteins, so proteins exhibit different type of structures: primary structure, secondary structure, tertiary structure, and quaternary structure. Out of all these, the enzymes are also made up of proteins, and those proteins which are present in enzyme, they are exhibiting the tertiary structure. So all enzymes are made up of proteins, but there is an exceptional case where enzymes are also made up of nucleic acids. So best example is ribozyme. So ribozyme is a nucleic acid, which is a part of which is an enzyme. So similarly, ribonuclease P. So P means here phosphate. So these are the two exceptional cases. So based on this question, so we'll be taking the answer as ribozyme. So option 4 will be our answer. So ribozyme is our answer in this case. So let us move to the next question. So mark the correct statement. That was the question. So most enzymes get damaged above 40 degrees centigrade. So no doubt some enzymes, so when they're heated above 40 degrees centigrade, certainly they get damaged. So no doubt about it. Statement 1 can be a right answer. Then rate of chemical reaction decreases by half for every 10 degrees change in either direction. So rate of chemical reaction, so decrease by half for every 10 degrees centigrade change in the direction. So that statement, so statement 2 is also correct. Then third statement, if you see, the enzyme carbonic anhydrase accelerate the rate of hydration of carbon dioxide by about 10 million times. So all these statements are correct, students. So statement 1, statement 2, statement 3, so all are applicable. So finally all these statements are correct. All of these statements are correct in this particular given question. So let's move to next question. So if you see this question, next question, if you see, so Km, so what is this? Km is expand. So Km is, if you see the Km is, Km is, how do we define Km? It is a Michaelis, So it is a Michaelis-Menten constant. So it is a Michaelis, Km indicates it is a Michaelis-Menten constant. It is a constant. So it is Km indicates Michaelis-Menten constant. So if you Km is defined as it is half the maximum velocity. So it is half the maximum velocity. So Vmax by 2. So Km is represented by one formula. The name of the formula is called Kmax is equal to Vmax by 2. So Km is half the maximum velocity. So Km indicates, so Km is a Michaelis-Menten constant. So what this Km measures? The Km measures the amount of substrate bind to the active site. So what is this actually the Km measures is? So Km calculates or Km, so it measures or it calculates. So it calculates or measures the amount of, so calculates or measures the amount of substrate. So amount of substrate, so measures or calculate the amount of substrate bind to the active site. So active site of enzymes, amount of substrate bind to the active site of the enzyme. So that's how the Km. So Km measures the amount of substrate bind to the active site. So that is what the Km. So Km measures amount of substrate bind to the active site. So Km, so what is the Km is an enzymatic reaction. So temperature, if you see that, so let us go to one by one options. So temperature in Kelvin, so this is a wrong statement. So statement 1 is a wrong statement. Then if you see statement 2, the value which all enzyme molecules are saturated, the value at which enzyme molecules are saturated, okay. Then the concentration of substrate, it is the concentration of substrate. Yes, so option 3 can be answered. It is the concentration of substrate. So it measures the amount of substrate, means it is measuring the amount of concentration of substrate binding to the active site. Okay, so option 3 will be our answer based on this question. So once again, what is this Km? Km is a Michaelis-Menten constant. So Michaelis-Menten constant. So Km explains, so how do this Km is defined? It is the amount, it measures the amount of substrate bind to the active site. So Km is half the maximum velocity, so it is Vmax by 2. So let's move to the next question. So the inhibition, so inhibition of succinate dehydrogenase by malonate. So inhibition of succinate dehydrogenase by malonate. Malonate is an example of. So succinate dehydrogenase. So where is this succinate dehydrogenase involved is actually, so in Krebs cycle. So in a Krebs cycle during the process of respiration. So succinate is converted into fumarate. Succinate is converted into fumarate. Succinate is converted into fumarate in the presence of enzyme. So name of enzyme is called as succinate dehydrogenase. So it is converted into succinate dehydrogenase. So succinate is converted into fumarate in the presence of enzyme. The name of the enzyme is called as succinate dehydrogenase. So succinate is converted into fumarate in the presence of enzyme. The name of the enzyme is called as succinate dehydrogenase. So this succinate dehydrogenase, so is inhibited by an inhibitor. So this succinate dehydrogenase enzyme, this is inhibited by an inhibitor. The name of the inhibitor is called as malonate. So this malonate is so it is inhibiting the, so it is similar to the structure of succinate dehydrogenase, and instead of succinate dehydrogenase, malate is going and binding to the succinate and stopping the formation of the fumarate product. So such type of inhibition is called as a competitive inhibition, or such type of inhibition is called as a competitive inhibition. So now we can take answer as it is a competitive inhibition. So option 2 will be our correct answer. So it is a best example of the competitive inhibition. So let's move to the next question. See the class of enzymes. The class of enzymes that catalyze. So that catalyze the removal. So catalyze the removal of group of substrate without addition of water, leaving double bond. So there is class of enzymes. So without involvement of water. So the enzymes, the best enzymes is lyases. So lyases do the same thing. So they remove the groups. So the removal of groups. So removal of a group. So removal of a group. So without involvement of water. So removal of group. So removal of group takes place. So without. So removal of group takes place without involvement. So involvement of water. So without involvement of water. So removal of group without involvement of water. So that leads to formation of double bonds. So that leads to formation of a double bond. So that leads to formation of a double bond. Such type of enzymes is called as a, such type of enzymes is called as a lyases. So the such type of members are called as a lyases. Next one. So prostatic group. So prostatic group of enzymes are called as. So the prostatic group of an enzyme. So prostatic group. So actually this prostatic group. So this is a of an enzyme. So prostatic group is a non-protein part. If you see the prostatic group. So this prostatic group, it is a non-protein part of the enzymes. It is a non-protein part. It is non-protein part of the enzyme. So it is a non-protein part of enzyme. And second thing, it is a cofactor. So it is a cofactor. So prostatic group is a cofactor, and it is a non-protein part of enzyme. It is a cofactor, and it tightly bonds. It tightly bonds to apoenzyme. It tightly bonds to apoenzymes. It tightly bonds to apoenzyme. So what is an apoenzyme? So protein part of enzyme is called as apoenzyme. So protein part of enzyme is called as apoenzyme. So what is an apoenzyme means? It is the protein part of the enzyme. So protein part. Protein part of enzyme. So protein part of enzyme is called as apoenzyme. So prostatic group. So it is a cofactor. In fact, the prostatic group is a cofactor. So it is a cofactor. Prostatic group is a cofactor, of course, and it is a non-protein part of the enzyme. It is a non-protein part of enzyme. It is tightly bound to apoenzyme. It is tightly bound to apoenzyme. So finally, our answer. So prostatic group. So best example of prostatic group is it is the heme. So heme is the best example of the prostatic group. And what is important point about prostatic group is it tightly bonds. It is a cofactor, non-protein part of enzyme, and it is a cofactor which tightly bonds to the apoenzyme. So that is how we define prostatic group. So option 3. So cofactor is our right answer. Next one. So mark the correct statements. So if you see the coenzymes NAD and NADP are the coenzymes that are derivative of vitamin B. Now this is the right statement. They are derivatives of vitamin B. Then catalytic activity is lost when cofactor is removed from enzyme. Yes. So no doubt catalytic activity, so cofactor is required for activity. So catalytic activity is lost when cofactor is removed from enzyme. Then, so in carboxypeptidases, the McLines are zinc. So carboxypeptidases, the McLines are zinc. So finally, all of these will be answers. All of these will be the answer. So mark the correct statements. NAD and NADP are the coenzymes which are derivatives of vitamins. Catalytic activity is lost when cofactor is removed from enzyme. So in carboxypeptidases, metalloenzymes are of zinc. So all of these will be our answer in this particular case. Let's move to the next question. 3. So 3 of the following enzymes are correct and 1 is wrong. So 3 of the following statements about enzymes are correct and 1 is wrong. So which one is wrong? So enzymes require optimal pH for maximal activity. So enzyme requires optimum pH. So every enzyme requires optimum pH for maximum activity. So that statement is correct. So every enzyme requires optimum pH. So optimum pH. So for example, if there is a pepsin enzyme is active at having its own optimum pH, that is 2 to 3. Then enzymes are denatured at high temperature, but in exceptional case there is an effective even at 80 to 90 degrees centigrade. So that statement 2 is also a right statement. So statement 2 is also a correct statement. Why? Because so if you see the members of bacteria like dermophiles. So dermophiles. So these are the group of bacteria which are called as dermophiles. So these dermophiles, they generally grow at high temperatures. So high temperatures. So generally grow at high temperatures, and average temperature generally ranges between 80 to 90 degrees centigrade. So best example is Dermis aquaticus. It is the name of the bacteria. So which is the best example is Dermis aquaticus. So dermophiles, these are the group of bacteria they can survive at high temperatures. So they can survive at an average temperature of between 80 to 90 degrees centigrade. So that is their importance, thermophiles. So that is the reason. So statement 1 is correct, statement 2 is correct. Then enzymes are highly specific. Yes, enzymes are certainly highly specific. So most enzymes are protein, but some are lipids. So that's what we are discussing now. All enzymes are made up of proteins, but some are made up of nucleic acids. We have seen some are made up of nucleic acids. So that is the reason statement 4 is the wrong statement. So wrong statement is statement 4. So option 4 will be our answer in this case. Let's move next question. So during protein denaturation, which of the following is disrupted? So we're discussing the same thing. So protein is, so we know that the enzyme, so the enzyme, which enzyme contains protein, that protein is having the tertiary structure. Now this is the tertiary structure which is having coiling and folding. So it is having coiling, so tertiary structure. Now it is having foldings, coilings and foldings. Okay, now this, when this tertiary structure, now this is a tertiary structure. So when this tertiary structure is treated with above, so when tertiary structure is treated above the 45 degrees centigrade or above 50 degrees centigrade, what happens is denaturation takes place. So denaturation takes place. So when the denaturation takes place, then what happens is they form simple structure like this. They lose all bonds and they form primary structure. So they form primary structure. So denaturation, whenever the protein gets denatured, the tertiary structure will be converted into the primary structure. Now according to our question, when protein is denatured, which of the following is disrupted? So which of the following is disrupted means, so the 3D structure is disrupted, because so the tertiary structure is disrupted and we form the primary structure. So that is the answer. So the 3D structure, the tertiary structure will be disturbed actually. So that will be our answer in this case. Let's move to next question. So which of the following statement regarding enzyme inhibition is correct? So let us see the statements. So competitive inhibition is seen when substrate competes with an enzyme. When substrate competes with the enzyme for binding to an inhibitor protein. So that is a wrong statement. So competitive inhibition is seen when substrate and inhibitor compete for active site. Now this is the right statement. So statement 2. So statement 2 seems to be most relevant. So statement 2 is the right answer. So in competitive inhibition, what happens is, so if we say this is an enzyme, so now this is the substrate. So this will be the substrate and round 1. Now think round 1 is the inhibitor. I represents inhibitor. Now this is the substrate. So enzyme. So in competitive inhibition, what happens is substrate will get compete with inhibitor. So instead of substrate, inhibitor is coming and binding. So they form complex, enzyme inhibitor complex. So they form complex, that is enzyme inhibitor complex. So like this, now this is called as a competitive inhibition. Now this type of inhibition is called as a competitive inhibition. Now this will be our right answer. So it is a option 2 will be our right answer in this case. So let us move to the next question. The catalytic efficiency of two different enzymes can be compared. So catalytic efficiency depends upon the Michaelis-Menten constant, that is Km value. So Km value. So catalytic activity depends upon the Km value. So option 3. So option 3 will be our answer in this case. So let's see this graph. So the given graph shows the effect of substrate concentration on the rate of reaction of an enzyme. So with what does this graph indicates? So substrate concentration, if you see the substrate concentration, okay, so if you see the substrate concentration, so if you see the substrate concentration, now this is the substrate. So now here on x-axis you have substrate concentration. So with increase in the substrate concentration, what happens is the rate of enzymatic reaction increases. So rate of enzymatic reaction is directly proportional. So you can take option 1 as answer. So in this graph, the graph is showing that with increase in substrate concentration. So increase in substrate concentration. So graph is showing that increase in substrate concentration. So graph is showing that increase in substrate concentration increases the enzymatic activity. So increases the velocity. You can see this increases the velocity. So that's what it is showing here. So increase in enzymatic reaction is directly proportional to the substrate concentration. More amount of substrate concentration, more is the enzymatic reaction is taking place. So that is the reason. So option 1. So option 1 will be the right answer in this case. Next question. So an organic substance bound to enzyme and essential for its activity. So an organic substance bound to an enzyme and it is essential for its activity. So that is called as. So it is called as holoenzyme. So if you take a holoenzyme. So what is a holoenzyme? So holoenzyme, it is having two components. Holoenzyme is having two components. So every holoenzyme has two components. So if you see what are the two components, so it has the two components actually. So what are the two components? So holoenzyme has two components. So it is having the two components, like it is having protein part and non-protein part. So non-protein part. So protein part and non-protein part. So protein part and as well as non-protein part. So let us write here. So holoenzyme. So holoenzyme has two main components. So the protein part, it has protein part. So the protein part of enzyme is called as apoenzyme. So it is called as apoenzyme. So the protein part is called as apoenzyme, and it is also having non-protein part. So enzyme has a non-protein part. So it is having non-protein part. That non-protein part is called as a cofactor. So this cofactor has organic cofactor and organic cofactor. So organic cofactor. So out of these multiple choice questions, if you see, so coenzyme. So coenzyme is an organic substance. So answer. So the cofactor, in that cofactor we have coenzyme. So coenzyme is an organic cofactor loosely bound to apoenzyme, loosely bound to apoenzyme. So based on that we take answer as coenzyme. So answer will be coenzyme. So coenzyme will be our answer in this case. So that is end of today's multiple choice discussion, students. So we'll see in the next class. So that is end of today's session, students. Thank you. So we'll see in the next class. So before closing this session, so once again I would like to introduce about our Unacademy Plus subscription, students. So if you see our long-term plans, so we have best long-term plans available. There is 6 months plan costing around 10,125 rupees and 12 months plan costing around 12,375 rupees, students. I suggest you to purchase this long-term plan. Certainly it will benefit for you. So thank you, students. See you in the next class.